Our first presenter is Kaavya Rathnakumar from South Dakota State University, and she's currently a Ph.D. student of the Dairy and Food Science Department. She will present on extraction of dairy phospholipids using switchable solvents.
Good morning, and thank you everyone for coming to my presentation. I'm really glad to be part of this AOCS ePoster pitch competition, and I'm really excited to present my work on extracting the valuable phospholipids, which goes as a waste in the dairy manufacturing stream. Phospholipids are naturally present in the milk fat, and during processing, they go into the byproduct. For instance, buttermilk from butter, beta serum from anhydrous milk fat, pro cream from cheese whey. All these contain an enormous amount of phospholipids. Why these phospholipids are of great importance, especially the milk phospholipids, are considered unique because of the nutritional and health benefits. Consuming these phospholipids helped in reducing cardiovascular disease, cholesterol absorption. In recent times, it is used in the infant formula for the growth and brain development of the infants, and is also used in various processing aids as natural emulsifiers. Since this pandemic, the consumer trends on eating more healthy and nutritious food has come along the way these days, and these byproducts serves this purpose naturally. However, the challenge lies in isolating these phospholipids. Since it is a very wet dilute material, it goes through a multi-step complex procedure since it has to be concentrated, centrifuged, fractionated, which involves a lot of time consuming and also use a lot of chlorinated and flammable solvents, which are injurious to health and is economically unviable. And they use a distillation process, which is also causes various environmental hazards. In order to mitigate these challenges, we came up with a smart alternative is the use of switchable solvents, which is based on reversible reaction of amines. Thus, the change in miscibility, it's from water immiscible, it can switch on and off from water immiscible form to water miscible form by simply bubbling the carbon dioxide. When you remove the carbon dioxide, it goes back to its original form, and the magic lies here. You can reuse the solvent for several extraction processes. From this technique, we were able to obtain 99% of phospholipids from the buttermilk compared to the traditional methods such as the Falch and the Merjonia method. And it was interesting to note that it was remarkably high yield, like fourfold higher compared to the any other traditional methods as well as techniques which are in existence. In order, however, the extraction efficiency depended on the type of the byproduct utilized in the switchable solvent extraction. So in case of the ultrasound, in case of the beta serum, in order to improve the extraction efficiency of the beta serum, an ultrasound pretreatment was employed prior to the switchable solvent extraction. And from this, we were able to obtain 70% of the phospholipids, which was 10 times higher compared to the beta serum, which was untreated. Further, this was applied into the ice creams, which was not depicted in my poster, which I did later on. And we were able to see 63% of phospholipids in the these phospholipids were mapped at every stage of the ice cream manufacturing process, and we were able to see interesting results in every stage of the processing, which I'll be working on this in future. And these phospholipids helped in converting a low value, this technique helped in converting a low value byproduct to a high value end product. In an economic point of view, this technique is highly beneficial as it is a sustainable and an eco-friendly way to extract the phospholipids from the byproduct source, and you can reuse the solvent, which makes it more sustainable. From this technique, we were able to and generate a higher extraction yield as well compared to any other techniques. And this project helps the dairy farmers in generating income by utilizing single drop of milk, and also it creates more business opportunities to industries by increasing the utilization of the dairy byproducts more effectively. In case of the ice cream, you obtain a converting a low value product to a high value end product, not only enriching it with ice cream with more nutrition, but also it's the phospholipids by itself serve as a natural emulsifier, and which I'll be working on in my future. Thank you.
Thank you, Kaavya. That was a great presentation. Judges, it's time for your feedback and questions. For those in our audience, please add your questions and comments in the chat. Judges, do you have any questions for Kaavya?
I guess I can go first.
Yes.
Great job, Kaavya. That was excellent.
Thank you.
Do you anticipate this approach could be used with other matrices?
So yeah, it can be used. This is a protein matrix. We are trying to disrupt this protein matrix and get the phospholipids. So similar way, it can be used for a carbohydrate matrix as well. It has been already studied, and so if it is so for the protein and carbohydrate matrix, we know it can be done. For other matrices, still studies has to be done to know like what type of switchable solvent will be used.
Okay, great.
I guess I'll jump in if you don't mind, Kaavya. That was a wonderful presentation. I'm very excited about your about your results. I have, I guess, two quick questions. You pointed out that you could reuse a solvent, which I think is a great result. How often do you think you can reuse a solvent, or is there a cap on how frequently you can reuse the solvent?
Okay, so theoretically, it is a reversible reaction, which is based on a Le-Chatelier principle. So it can be used several times. Like it can switch back and forth several times by simply bubbling the carbon dioxide and removing the carbon dioxide. But however, it has to be. It depends on the concentration of the lipids, or like the different different material concentration, like some other minerals might have in the influence, but we haven't studied that. But theoretically, it can be used like several times.
And does efficiency change as you reuse it?
Yeah, it might change. As I said, it might change depending on the concentration of the products.
Okay. And my other quick question is, how long is this extraction process altogether?
So it is around, it depends, like from 3 hours. We have tried from 3 hours to 18 hours.
Okay. And is like the efficiency or the amount that you're able to extract, does it increase monotonically? Like with more time you extract more? Okay.
So we were when we used a temperature, we were able to obtain high at 60 degrees Celsius with time like 3 hours. But however, it depended on the type. Still more optimization has to be done, and to know like the exact time to get a very higher yield. But with 3 hours, we can get a higher yield.
Great.
Kaavya, this is Sneh, and great presentation, quite applied, and congratulations for nice piece of work. My question to you is, one thing which I observed in your results of the phospholipid extract from the buttermilk serum, I feel there is an enrichment of the phosphatidylinositol fraction in your phospholipid extraction and the extract. Do you know why it may be? Is it the nature of the way it is extracted? And then I'll ask my next question once you have opportunity to answer this.
Okay, so yes, so since this phospholipids are present in the outer and the inner membrane, we studied a mechanism study to know like how the switchable solvents interact with the inner membrane and the outer membrane of the phospholipids. This phosphatidylinositol or the other enzymes which are present in the inner membrane, and the switchable solvents had an ability to go inside the membrane and extract these phospholipids, which is based on an ion pair formation. And so from that we were able to know that this linositol was higher concentration of phosphatidylinositol was there, like from our 4 different studies, like all were like the linositol was higher. But however, the presence of sphingomyelin and also other individual components like phosphatidyl, ethanolamine, choline were also present, and the distribution was studied using an HPLC, using charge aerosol as a detector, and yes, we were able to get that.
And my last question will be, if that's the case, that the enrichment phosphatidylinositol, and as you told at the onset, the phospholipids concentrated or isolated by this way may be useful as one of the health benefit ingredient. So what do you think will be implication if it is the phosphatidylinositol is enriched?
So as a whole, it has more nutritious benefits, but then we haven't studied the bioactivity, which can be done in future, and also like we are still working on the ice cream, like what are the individual phospholipids present, quantifying them at each stage of the ice cream manufacturing process, so that will give a more clearer idea on like what type, and also studying the bioactivity. But overall we know that have like anti-inflammatory properties, other immunological responses, and also it is used in the liposomal production of liposomes. So it's like overall we know that, but still we had to do the bioactivity to know how much it can be obtained from that. Thank you. I think you are mute.
Yeah, I mean, thanks a lot. Appreciate it.
Thank you.